The transcribing of the nitrogenous bases or the sequences that are present on the DNA onto the mRNA strand. So in prokaryotes it is different, in eukaryotes it is different. Anything and everything or all the metabolic activities in our body are regulated because of these genes. Hello everyone, a warm welcome to this session for class 12 biology. I'm Dr. Divya, biology faculty. So in the session of this chapter, that is chapter 5, molecular basis of inheritance, we will learn about regulation of gene expression and the lac operon concept. So in the previous session, we had learned about the translation mechanism, right? So in one of the sessions, we had dealt with transcription, then we talked about translation, and now how the genes exactly gets expressed after the translation mechanism we are going to look into. So to briefly brush up, so if you need in detail, you can watch those two sessions related to transcription and translation. For you to understand now what it is, we'll just brush up. So transcription is nothing but the transcribing of the nitrogenous bases or the sequences that are present on the DNA onto the mRNA strand. So in prokaryotes it is different, in eukaryotes it is different. In prokaryotes it's a simple form, eukaryotes the mRNA strand will contain both introns and exons. Therefore the introns, which are the non-coding sequences, need to be removed, and for that particular process it undergoes capping, splicing and tailing mechanism. So that we have studied. Now the mRNA strand is there, it is ready, it is containing only the coding sequences or nitrogenous bases which codes for the genes, and those will move into the cytoplasm. Next, moving to, after transcription, translation. Now whatever genetic code which is in the form of triplet is present on the mRNA, that should be read, that should be transcribed. So for that there is the tRNA molecule or the tRNA which contains the anticodon region, which will come and bind to the respective coding sequences that are present, or the triplet codes that are present on the DNA, and the amino acid, chains of amino acid goes on synthesizing from the promoter till the terminator region. All that we have studied. And meanwhile ribosomes also take part, because those amino acids need to be converted into proteins, or because at the end of the day proteins are nothing but chains of amino acids, and it is produced by the ribosome. So the ribosome, both the large unit and the small unit of the ribosome, they participate in this particular process, wherein at the end the protein gets synthesized or chains of amino acid gets formed, and those they come into the cytoplasm, and then whatever ribosomes is there, it will get detached and it will go back into the, and will remain in the cytoplasm to perform its next role of protein synthesis. So all that we had studied. It is just a brief that is description I'm giving you for you to brush up. So if you need in detail, you can visit the previous sessions of this chapter. So now we know this one, so we'll move on to learning about regulation of gene expression. So how the genes get expressed? Now, do you think that the genes get expressed throughout every time in our body? No. There are some times when the genes remain switched off, and sometimes when the gene remains switched on. So when they get switched on, then the gene expresses itself producing this, or expresses itself involved in the process of this transcription or translation mechanism. So let's talk about it. So in eukaryotes, the regulation of gene expression takes place in the following level. First one is the transcription level, wherein the formation of the primary transcript, that is the formation of the mRNA, will take place. You have learnt about that. You can visit the previous sessions to have a look at it. Next is processing level. So processing level is regulation of splicing. As I told you, splicing mechanism, that is whatever introns are there, they need to get removed from the coding sequences. So they need to get removed. So it will have introns, exons on the sequences, introns and exons. So these needs to get removed. So that particular process we call it as splicing. Then regulation of splicing will take place. Then after that, the mRNA that is there inside the nucleus itself, now we call it as the nascent mRNA or the young mRNA, which contains only the coding sequences, that will move from the nucleus into the cytoplasm of the cell. Then it will move into the translation level. I've told you, right, how the reading of the mRNA strands or the transcribing of the mRNA strand takes place with the help of the tRNA as well as the ribosome. So, all these 4 steps are involved during the regulation of gene expression. Now, it is time for the genes to get expressed. How? We will study taking an example. So, the genes in a cell are expressed to perform a particular function or a set of function. Anything and everything or all the metabolic activities in a body are regulated because of these genes. So, at a particular time, whenever the genes for a particular activity or a set of activity needs to get expressed, it needs to get expressed. Otherwise, all the metabolic activities or the respective metabolic activity which was supposed to be done by that particular gene will not take place properly and lead to a lot of disorders in a body. So, therefore, the genes in a cells, they need to get expressed at regular times or at a particular time in order to perform a particular function or a set of functions. So, we'll look into it taking an example. So, for this we can take the lac-operon concept as an example. In detail, I'll explain to you about lac-operon concept in the coming slide. So, you'll understand that clearly. So, here example, if an enzyme, say for example, called beta-galactosidase enzyme. So, this beta-galactosidase enzyme is synthesized in a bacteria which is called as Escherichia coli. And this beta-galactosidase enzyme that is there, which is synthesized by Escherichia coli, it is used to catalyze or it is used for a particular function. What is that function? That function is hydrolysis of a disaccharide. So, disaccharide is nothing but lactose. So, what it does? It converts lactose. It converts lactose into galactose and glucose. Which enzyme? Beta-galactosidase enzyme. So, this beta-galactosidase enzyme, which is produced by E. coli bacteria, in that particular E. coli bacteria, its function is to convert lactose into galactose and glucose. Lactose is all these are disaccharides or galactose into glucose. So, it converts the disaccharide into galactose and glucose. And the bacteria uses them as a source of energy. We know that, right? Any organism, the source of energy is glucose, be it plants, be it microbes or humans, right? And that glucose is the one that gets converted into energy. In case of plants and in case of animals, how it gets converted into energy, you have studied in your first PUC and your class 11th. You're quite familiar with that, right? In the process of digestion, in the process of photosynthesis in plants, digestion in animals, and also during respiration also. So, this is what it does. So, now we are talking about E. coli. So, E. coli bacteria contains an enzyme called as beta-galactosidase. That beta-galactosidase enzyme helps in the conversion of a disaccharide called lactose into galactose and glucose. And this galactose and glucose is needed for various metabolic activities to happen properly in the bacteria, because for all these metabolic activities, they are the source of energy. So, now we know about this. Now, hence, if the bacteria, say for example, from where will this lactose or from where will this beta-galactosidase come? It's an enzyme, right? And enzymes are also nothing but proteins. And at the end of the day, proteins are nothing but chains of amino acids. So, therefore, they need to be produced by the triplet codes or the genetic codes or the genes that are present on the mRNA strand, mRNA sequences, right? You've studied that in translation mechanism. So, here now we will look into, so just remember what we have, beta-galactosidase enzyme. Beta-galactosidase enzyme is needed for conversion of lactose into glucose and galactose, which can be used as energy source by that particular organism for various metabolic activities. Now, if that particular bacteria, that is E. coli, say it doesn't have lactose in it. So, if it doesn't have lactose, so if that bacteria doesn't have lactose, then from where it will get energy? Of course, you can say it gets energy from glucose and galactose. But from where do the bacteria get glucose and galactose? It is from lactose. So, the bacteria needs lactose. Now, let's assume, say for example, the bacteria doesn't have lactose. So when the bacteria doesn't have lactose, they will not produce any energy that is required for the metabolic process. So hence, if the bacteria do not have lactose around them to be utilized as energy source, they would no longer require the synthesis of the enzyme beta-galactosidase, right? Beta-galactosidase is synthesized in the body of the E. coli because it has a work. What is the work of the beta-galactosidase enzyme? To convert lactose into galactose and glucose. Now, if that lactose is itself not synthesized in the body of or inside that particular E. coli bacteria, then what will be the function of beta-galactosidase? So, therefore, beta-galactosidase will also not get synthesized. So, therefore, in simple terms, it is the metabolic, physiological or environmental conditions that regulate the expression of genes. So, that is what it is. So, not just the genes are regulated because of the mRNA strands or because of the mechanism that happens inside a body. External factors, the environmental factors, are physiological condition, and also the metabolic conditions of a person or any other organism can affect the synthesis of a particular enzyme or a compound. So therefore, for expression of the genes, all these metabolic, physiological and environmental conditions also play a major role. So, if the bacteria, because of metabolic or physiological or environmental conditions, the bacteria is not capable of synthesizing lactose, or the bacteria doesn't have lactose in its body, then therefore the gene will not, the respective gene that is there which is supposed to produce beta-galactosidase enzyme, it will not produce because lactose is itself not there, so therefore it will not get produced. So therefore all these factors are also important for gene expression. We'll take one more example, that is the development and differentiation of embryo into adult organisms. So embryo. So how does embryo form in any of the organism, be it animals or in human beings? If I take an example, the male and the female gamete which are haploid, they fuse together to form a diploid zygote, right? That zygote, diploid zygote, then differentiates to form an embryo, and that embryo differentiates to form the fetus, which has its limbs, the digits, the head and all that region, right? So the development and differentiation of the embryo until it becomes an adult organism, all those development are regulated by the gene expression itself. So whatever, say for example, if I take even the texture of your hair, how your nail, with some of them will have shiny nails, some of our nails will look very dull, some of us will have soft nails, some of us will have brittle nails, right? So that and all depends on a protein which is called as keratin. Now if that itself so how much amount of keratin, what quality of keratin is produced by the gene, that actually results in the condition of a nails or hair and all that. So that is what. So right from the development of the embryo until the adulthood, any function that happens in the, or metabolic function or physiological function, anything that happens in the body of the organism, everything is controlled by the gene expression. So therefore, it is very, very important for the genes to get expressed at a particular time. And what we eat, what kind of environment. So the expression of the genes can also get altered because of the polluted environment when we are exposed to too much of pollution and all that. So all that is about the gene expression. So these are how the genes get expressed. So these are some of the examples that we have taken. So moving further, in prokaryotes, so regulation of gene expression in the case of prokaryotes. In prokaryotes, control of the rate of transcription initiation is the predominant site for control of gene expression. So control of the rate of transcriptional initiation. How many times the transcription initiates or begins, that many times the genes will get expressed, because the genes need to be read, and when the genes are being read during the process of transcription and translation, at the same time the genes will get expressed also. Genes getting expressed means genes producing chains of amino acids and that leading to the formation of the protein. So that is what it means. And in a transcription unit, the activity of RNA polymerase. So RNA polymerase enzyme, it is a DNA dependent RNA polymerase. So during the transcription process itself, when the complementary mRNA strand is being formed for the coding strand of the DNA to one of the strands of the DNA, during that time itself the genes should be, or the sequences should be read, and there should not be any mutation, be it point mutation or shift mutation, anything should not happen. If it doesn't happen, and if all the genes are present or all the sequences are in position during the transcription process, right from the initiation up to the terminator region, then the gene expression will be perfect. So that is what it means. So therefore, in a transcription, and RNA polymerase enzyme also needs to get synthesized every now and then, because it is that enzyme that keeps on adding the DNA sequences to the mRNA strand. Now, if RNA polymerase doesn't go and bind to the initiator region and the process doesn't happen properly until the terminator, then proper addition of the sequence on the mRNA strand will not take place. Therefore, the mRNA strand can completely get mutated. So that is why in a transcription unit, the activity of RNA polymerase at a given promoter or initiator region in turn is regulated by the interaction with the accessory proteins. Apart from that, there are some accessory proteins or some sigma factors or some factors which are there which also assist in that particular region for the RNA polymerase to recognize the promoter region. So if you remember in the previous session in the sense, I'm talking about the transcription wherein we learnt about transcription. In that session, you had seen that sigma factor, right? So how will the RNA polymerase enzyme know where exactly the promoter region is there? It is because of the accessory protein. Say for example, sigma factor. So those accessory proteins, it acts like a map or a navigator which tells the RNA polymerase, okay, this is the region where the promoter region is present or promoter sequence is present, go and bind there. So that is what it means. So therefore, all the activities on the sequence needs to be in a proper fashion. So these regulatory proteins or accessory proteins which regulate the gene expression through transcription and then through translation can be both positive or it can be negative. So do you think that in the genes every time there are only genes which get activated? No, there are only proteins which can activate the genes. No, sometimes some of the genes for the good it needs to be switched off. So it should not get expressed. So at that time there are some proteins which are called as repressor proteins. So these repressor proteins, if it goes and binds at the promoter region, no, then the regulation of the gene expression will not take place. If they don't go and bind, then the regulation of the gene expression will take place. So these regulatory proteins, sometimes they can act as activators which activates the gene regulation, or they can act as repressors which inhibit or repress the gene or suppress the gene expression. So we'll study about that. So the accessibility of promoter region of prokaryotic DNA is in many cases regulated by the interaction of proteins with sequences which are termed as operators. So there are some operator regions or promoter regions, especially operator regions which are present. So if the repressor protein goes and binds, you'll study about that in lac-operon concept. Okay, you'll be clear about that. Now I'm just telling you how gene expression, what all are important for gene expression. So for gene expression, activator proteins as well as repressor proteins participate. So there is a region on the, so right from the promoter region, we have the promoter region, then we have the structural genes. And if you remember, in the previous session we had learned, right, there is a promoter region on the DNA strand, then comes the structural genes, and then comes the terminator region, right? The reading of the strand starts from the promoter until the terminator, it will take place. Now in between, before the structural genes, there is a site which is called as the operator region. Now if a repressor protein goes and binds at the operator region, then the gene expression will not move further, or it will stop. And if the repressor doesn't go and bind to the operator region, then the gene expression will continue. So that is what it means here. So the operator region is adjacent to the promoter elements in most operons or in most sequence. And in most cases, the sequence of the operator bind a repressor protein. So repressor protein, if it goes and binds, then the gene expression will not take place. So each operon has its specific operator and specific repressor. So entire strand, they will have specific operators and specific repressors, wherein the repressor or the operator proteins can go and bind. So we'll learn about that in detail, wherein when I talk about lac-operon concept, you will understand this. So lac-operon concept, we'll learn about this. So this is taking the example of prokaryotes. So while talking about gene expression, if you remember, I had taken the example of E. coli, right? So wherein E. coli, we took the example of beta-galactosidase enzyme, and beta-galactosidase enzyme, its main function is to convert lactose into glucose and galactose. So the same thing, how exactly it happens, we shall look into. So we'll take the example of prokaryotes. Lac operator is present only in the lac operon. So I told you specific sequences have specific operators. Now because we're talking about the lac operator. So lac operator. So this is the lac operator region. Operator. So if you can see here, there is the promoter site. There is the regulatory gene or the inhibitor gene is present here. Promoter region is present here. Regulatory or inhibitor gene is present here. Then again the promoter region is present. Then comes the operator region. Then the Z, Y and A, they are entirely structural genes. After a stretch of structural genes, then comes, after a certain stretch, then again maybe there might be again a operator region again. It might code for another enzyme. It's like that, okay? Here we are talking about the lac operator. So in lac operator, moving back, so lac operator is present only in the lac operon. That is in the sequence of DNA strand that synthesize the enzyme that is needed for converting glucose and galactose. And it interacts specifically with lac repressor only. So the lac repressor. So lac repressor is a protein which prevents or which goes and binds to the operator region. So, and therefore if it goes and binds to the operator region, gene expression will come to a halt or it will come to a stop. So here lac operator is present only in the lac operon and it interacts specifically with what? Only the lac repressor protein. Now in this there are other regions also which are present. I is nothing but the regulator gene or its inhibitor gene which codes for the repressor of the lac operon. So from where do we get this repressor protein? As in such, it can't be produced in the cell just like that, right? Again, the gene should be responsible for producing this particular protein. So the protein is produced by this particular inducer or inhibitor. So it is produced by this particular inhibitor, or we call it as the regulatory gene. So this regulatory gene I, it is also called as the inhibitory gene because it codes for the production of the lac protein which goes and binds to the lac operon or the lac operator region. So that is its function. So I is regulatory gene which is also called as inhibitor site that codes for the repressor of the lac operon. So the repressor, that is the lac operon repressor or the lac repressor, is produced by what? It is produced by the I gene. Remember, like that. Next comes the structural genes. These, the structural genes Z, Y and A. So Z, Y and A are the structural genes. In that, the Z structural gene codes for beta-galactosidase. So Z codes for beta-galactosidase. Now again brushing up what all we have now. We have the promoter region where actually the transcription begins or the translation begins. Then comes the inhibitor region or the regulatory gene, that is I. The function of the regulatory gene is to produce a repressor protein. So then comes the lac operator site and then comes the structural gene. So in the structural genes, the function of the structural gene Z is to synthesize the enzyme beta-galactosidase. Next. And we also studied that beta-galactosidase is important for what? To convert lactose into galactose and glucose, right? So therefore, all the things we have here now, we have the lac operator, then comes the gene that produces the repressor protein. Then we have the Z, the structural gene that produces beta-galactosidase enzyme. Then moving to the Y gene. Y gene codes for permeases. There is an enzyme which is called as permeases. So what does Y gene produce? It produces an enzyme called as permeases. What is their function? We will look into. So Y gene codes for permeases. Then we have one more gene. So Y gene codes for permeases. What is its function? So permeases is an enzyme which increases the permeability of the cell to beta-galactosidase. Whatever beta-galactosidase is produced by the structural gene Z, that needs to be absorbed by the cell. So the permeability of the cell to absorb beta-galactosidase will be increased by the permeases enzyme which is produced by the structural gene Y. Moving further, what is the function of the A gene? A gene encodes a protein. It produces the enzyme called as transacetylases. So what they do, we will look into. So now we understood P, promoter region, I, inhibitor gene which produces the repressor protein. Then O is the operator region where the repressor protein will go and bind. That is, lac repressor protein will go and bind. So therefore it is called as the lac operator region. Then we have the structural gene, that is Z, which produces the enzyme beta-galactosidase. We have the structural gene Y that produces the enzyme permeases that makes the cells more permeable to absorb the beta-galactosidase enzyme. Then comes the A, structural genes, that produces the enzyme transacetylases. Now we'll see what will happen now in the absence of an inducer. So inducer actually regulates the gene expression, or inhibitor actually stops the gene expression. So now we need an inducer. So that inducer is nothing but glucose. So now we need an inducer. So that inducer is nothing but what? It is lactose. The inducer is the lactose. Now, in the absence of lactose, what happens? Say for example, there is a repressor mRNA that is being synthesized by, so this is the repressor region, right? So I is a repressor region. So obviously for that, during the process of transcription, a repressor mRNA will get produced. So the genes which are repressor in nature, that will get produced. Now, so that repressor mRNA will produce the repressor protein. Now this repressor protein, it will go and bind the lac operator region. So when it goes and binds the lac operator region, will the further transcription process take place? Will the further reading of the strand happen? No. If further, or will the further translation mechanism take place? Now, if further translation mechanism doesn't take place, then how will the beta-galactosidase enzyme be produced? Or how will the RNA polymerase move further to read the strands? All that will get stopped. So, therefore, the gene expression will not take place. And next, moving further, so repressor binds to the operator region and prevents the RNA polymerase from transcribing the operon. Now, if these strands doesn't get read, then how will the genes be switched on, or how will the genes get expressed? So, therefore, it will prevent the expression of the genes, and therefore, beta-galactosidase will not be produced. And if beta-galactosidase will not be produced, then lactose cannot get converted into glucose and galactose. Next, moving further, in the presence of inducer. Now, if an inducer is present, that is, if lactose is present. So, if lactose, so that is how here in the E. coli, lactose is not there. Lactose is not there because the lactose inducer is not there. The lactose inducer is not being produced. Instead of that, the repressor protein is being produced. The repressor protein will go and bind to the operator. Therefore, beta-galactosidase will not work. Remember, previous slides we had learnt about, or beta-galactosidase will not get synthesized. Same here. Now, if inducer is present, that is, if lactose is present. If lactose is present, what happens? The repressor mRNA will do its function. It has to do, because the function of this region is to produce the repressor protein. So, repressor mRNA will get formed, transcription will happen, repressor mRNA will get formed, that repressor mRNA will produce a repressor protein. That repressor protein now, because an inducer, that is lactose, is already present inside the body of the E. coli, that inducer that is there, it will go and bind to the repressor protein. Now, when it goes and binds to the repressor protein, the repressor protein will become inactive and it will not be able to go and bind to the operator region. Now, the operator region is free, and therefore, now the RNA polymerase enzyme that is there, it will start reading the strand. When it reaches here, it will start producing the mRNA, which is lac mRNA. So the lac mRNA will get produced and beta-galactosidase enzyme will get produced, permease enzyme will also get produced, and transacetylase enzyme will also get produced during the process of transcription or during the process of translation. Here translation, it will not take place at all. Why? Because already the inducer has gone and bind and further steps will not happen at all. But in this case, the binding of the repressor to the operator region has been prevented by an inducer, which is the lac inducer. So that is what we, or because of the presence of the lactose. So this is how the lactose gets converted. So now this enzyme is also being produced, permease is also being produced. The bacteria cells can now easily take in the beta-galactosidase and therefore it can convert the lactose into glucose and galactose. So that is how the switching on and the switching off of the genes will take place. Here the genes will get switched off. Z, Y, A all will get switched off. Why? Because further expression itself will not take place because the operator region is bound by a repressor protein. Here the genes will get switched on because the inducer is preventing the repressor from going and binding to the operator region. Therefore, further reading of the strands by the RNA polymerase enzyme will take place and the mRNA strand will get produced and the mRNA strand will undergo transcription, that is translation mechanism, and it will produce the respective enzyme. So this is about, so this is what keeps on happening in all of entire length of DNA sequences. So this is about the lac operon concept. So to understand this, you also need to learn transcription and translation. You have to be thorough with that also. So this is about lac operon concept. So this was the session wherein we learnt about how the genes get expressed, and we learned that by taking the specific example of lac operon concept. So in the next session, we'll talk about human genome project, that is how our entire genome was or DNA was sequenced, and we learn about a technique which is called as technology which is called as bioinformatics. So see you in the coming session with these two topics. Thank you.